Very much. So I'm very happy to be here. I have a presentation. I'm used to give presentations, but not this type of presentation. So I was not exactly sure what to present to you. And so I want this to be very informal. And please feel free to interrupt me and ask questions when you feel like you want to know something. So this is the campus, the southeast corner of the UF campus where the Health Science Center is. This is a little bit of outdated picture because now there's an additional 2 towers here of the hospital that is being built here. But the Shands Hospital is here, and this is the Health Science Center with the College of Medicine, and this is called the Dental Tower. And my lab is in this big, old, ugly 11th floor building without any windows. So my lab is on the 6th floor here of this dental tower. So this is the outline of what I'm going to talk about. So first I'm going to give you a little introduction to tell you who I am and what I do. Then I would like to spend a few slides to talk in general why I think we need science literacy. I think it's a very important topic, and it's even more important in the past few months where we are hearing some interesting versions of what facts and alternative facts are. So I would like to spend a few slides on this. Then I would like to give you some ideas why you should consider doing undergraduate research project. And then I'm going to talk to you a little bit about what type of work we do in my lab. First, I'm going to introduce what lupus is as a disease, and then give you some ideas about the kind of things that we're doing in the lab. And again, you can interrupt me whenever you want. All right, so I come from France, and I grew up in the south of France here. And my dad was a farmer. My mom was an elementary school teacher, so I grew up in a small farm. It's not all as pretty as that, but it's very pretty. And this is lavender, and I love lavender fields. Then I went to school to Marseille. So Marseille is a big, see, so not very far. In France, you go typically to graduate school or to undergrad and graduate school to the nearest university that there is to where you live. So I went to school in Marseille, which is right here. It's a big port city. It's a big melting pot on the Mediterranean. And so I did my undergrad, and I got my PhD there in neuroscience. Then I came at UF in 1985 for 1 year as a postdoc fellow. So postdoc is a training that you do after you have your PhD, but you're not quite ready to do independent research. So it's called postdoctoral fellow. And I've been at UF ever since. And I'm currently a professor, as Beatrice was saying, in the Department of Pathology. And the full title of the department is Pathology, Immunology, and Lab Medicine. So this department is a combination. It's a big department. There are, I think, close to 400 members in the department. And part of the faculty in the department are pathologists. They are physician pathologists. And what they do is help in the diagnosis of diseases. So for instance, if you have a skin cancer, the dermatologist is going to take a biopsy, and then the pathologist is going to determine if you have a skin cancer or not and what kind of cancer you have. So those are faculty taking care of patients. And then there is a group of us doing research in pathology, and this is what we call experimental pathology. So we are trying to understand the basis and the mechanisms of disease. So this is what we do in general as research. So you probably don't have a good idea about what it means to be to have the job that I do. So what do I do at work? So I direct a research program. I don't do a lot, very little, in fact, of experiments at the bench. I've done that a lot in the earlier years of my career, but right now, I typically tell people what to do. I don't do it myself. And most of the times, they know how to do things much better than I know how to do it because I haven't done it in a long time. I read papers a lot to know what other people are doing in my field. And so this is a very important part of research. And this is something I like a lot because I like to learn. And things are always different. And sometimes you're mad because somebody else has done something that's very clever and you would have liked to do it yourself, but somebody beat you to it. But it's important to know what other people are doing. And things are moving very fast. I also design experiments, conduct analysis, coordinate work with collaborators, coordinate work of people in my lab. I also write papers and give talks to present our results. So once we find interesting things that are enough to package together, then we, as part of the work that scientists do, is to disseminate the research results, right? So we have to write papers that we submit to scientific journals. And most of the time, so those papers are peer-reviewed, and we'll come back onto that. And then they ask you to change things, and then you do additional experiment, and then it's published. I also spend a lot of time writing grant proposals to fund my research because we do not receive fixed set amount of money. All the money that is funding my research, I have to fight for it. And we have to fight very hard to get money for research. I'm also responsible for compliance. So there are a lot of rules and regulations in conducting research, and I'm responsible for that. I'm responsible to know all the rules, to take the training, and then to make sure that all the people in my lab know all the rules and are also compliant with the training. If I mess up with this, my funding can get cut. So this is a very important part. And also, I manage my lab. So I have a budget, and I have more or less 10 people in my lab. I'm responsible to pay their salary. So it's almost like being a small business owner, which is not something that typically scientists are very good at to start with, because otherwise we would have been doing business. But this is something very important. If you don't know how to manage your money, then you get in trouble. I also teach graduate students. I teach graduate courses in the classroom. I also do a lot of one-to-one mentoring with students in my lab to teach them how to do research. So learning how to do research is a very long process. It takes many years to not only learn the benchwork techniques, but also to learn how to design experiments, to do analysis, to interpret the results, to write your results, to read the literature. So it takes a lot of one-to-one teaching to get to that point. And I also do administrative work as a director of the division. So this is not really research. All right, so why do we do science literacy? So you probably know this guy. He's a popular guy, Neil deGrasse Tyson. He's a brilliant astrophysicist who has a gift for communication. And this is his word: science literacy is a vaccine against the charlatans of the world that would exploit your ignorance. So I think that even if you're not a professional scientist, as I am, everybody should have a minimal amount of understanding about how science works, because you need this to make very important decisions in your life. The benefits of science are many, and I like that. This is just satisfy curiosity. I've always been curious, and most of us scientists, we just do this because we love it. We just want to understand things, even if it doesn't have a direct benefit, but most of the time it does. And so this is many aspects of why science in general should be an integral part of our society. So what do we want from scientists? I just picked that from Facebook in the last few days. What do we want? We want evidence-based science, and we'll get to what it means. And when do we want it? After peer review. So these are very important concepts, right? So this is evidence-based science, and I moved to medicine because this is my field. So what is evidence-based medicine? Evidence-based medicine is an approach to practice medicine to optimize decision-making by using evidence of well-designed and well-conducted research. So it's basically saying that there are facts and then that are based on well-conducted research, and we're not going to have time to go into how well-conducted research is designed. So there are facts and there are opinions. And evidence-based science is based on facts and not on opinions. And then, so this is the first level of evidence-based science or medicine. It has to be facts acquired in well-designed experiment. And then it has to be peer-reviewed. So peer-reviewed, it means that once you have your results, then you present your results to your peers. And then they review and say, well, this makes sense or this does not make sense, right? And this is when something is accepted as a fact in science. So I'm going to give you just a few popular examples, right? So you're all aware of examples of when science and popular belief collide. So one of my favorite topic is vaccines. So I'm an immunologist, so I'm a strong believer in the value of vaccines. Vaccines have been one of the greatest achievements of modern medicine. And as you're all aware of, there's still an interesting debate on vaccines being the cause of autism. And this was based on the fact that in some cases, some kids get vaccinated and then they develop autism, right? And there have been a large number of well-conducted studies that have shown that there is no causative link between being vaccinated and getting autism, right? Sometimes it occurs together, but there's not a relation of cause and effect between the two. Another topic, which is a little bit older, was silicone breast implants causing cancer. So, it makes sense. You introduce something in your body, and then some women develop breast cancer, and so there was a popular belief that silicone caused breast cancer. And as a scientist, there is no reason to think that yes or no. But there were a number of trials that were conducted and studies that shows that there is no relationship between having a breast implant and developing breast cancer. So then the US government lifts a ban on the breast implants because there was no cause and effect between the two. A more recent one was on arthritis. So arthritis is results from the overuse of joints in older people, and it's because some of the components of the joints become go away, and then there is rubbing on the two parts of the joint, and it causes pain. Two components of the joints are glucosamine and chondroitin. So it makes sense that if you take supplements of chondroitin and glucosamine, then it would go to your joints and you would feel better, right? And even it's a very popular supplement for dogs, right? For all dogs, you give them. So a number of studies have been conducted and published recently by NIH that I forgot that there is no pointer. There is absolutely no evidence that those supplements have an effect. This is really big business. People are making a lot of money selling those supplements that are still being bought, and people are still taking them, but there is no evidence that those supplements are causing any benefit for patients. So these are a few examples of why we need science literacy, because you need to be informed to be able to make decisions. If you still want to take those supplements, that's up to you. It's not going to do any harms except to your wallet. But you have to know that it's probably not going to help a lot. All right, so enough from my little pulpit here about science literacy. So why should you consider to do a project as an undergraduate student in biomedical sciences? So first, you have to be curious. You have to love to learn. If you're not curious, if you don't try to understand things, if you don't like to solve mysteries, it's probably not going to be for you. You have to be interested in health-related issues. That's probably most of us are. You have to like biology. Very importantly, you have to be meticulous. If you're very sloppy, if you don't follow directions, this is not going to be for you, right? Because if you have 10 tubes in front of you and you have to label them with patient numbers or condition numbers, if you mess that up, then your entire thing is going to be messed up and it's not going to be useful. So you have to be meticulous and follow instructions. And very important too, that I tell to all my students, when you mess up, everybody messes up, right? When you mess up, you have to recognize that you mess up and say, I messed up, right? I'm going to get it. I have a cold, I'm sorry. I'm going to take a sip of my tea. And also, the last thing is that you have to work well with others. So a lab is a little close-knit family, and you have to work with other people. And if you kind of steal other people's reagents, if you take all the medium from somebody, you don't replace it, or do things like that, you're not going to be very popular in the lab. So you have to be considerate and work well with others. Sorry, I'm thinking I'm going to take a throat. So as all parents, I have 2 sons, but I have had graduate students, and we are very proud of our grad students when they do well. So I'm going to tell you in 3 slides the story of my first grad student, Kim Blemen, who came from Barbados. Her parents were working in Citrus Grove in South Florida. And she started in my lab doing undergrad research. So she did undergrad research in my lab for a couple of years. Then she graduated from UF, joined my lab and got a master's degree in lab medicine. Then she stayed in my lab to get a PhD degree in immunology. She was hired as a staff scientist by Procter & Gamble in Cincinnati to do research on autoimmune disease. While she was working at Procter & Gamble, she started her own consulting company for clinical trials in autoimmune diseases. Then she was hired as a postdoc fellow, then an assistant scientist at City of Hope in LA, which is a major cancer hospital. There she developed a unique imaging tool to track the progression of certain tumors, and based on that, she was hired 2 years ago as an assistant professor in pathology at Yale Medical School. And for those of you who don't know, Yale is medical school is considered one of the very top Ivy League medical schools in the nation. So I'm very proud of Kim. We see each other every year at professional meetings, and I'm very proud of her. So this is a picture of my lab, just to show you what I was talking to you about earlier, about a lab is a close-knit group of people working together. So some of the people have left and we have new people, but this is typically what a lab looks like. So we have people from different countries. Usually, well, I'm the oldest one, so we have younger people working in the lab, and the goal is to make everybody working together. All right, so now we're going to talk about lupus. So what is lupus? So this is in the world of lupus patients. I found that on a website of the Alliance for Lupus Research, which is one of the major advocacy groups for lupus patients. So it's a systemic autoimmune disease. So autoimmune means, in the world of this patient, I'm so awesome, even my immune system can get enough of me. So this is the immune system attacking your own body. Systemic means that it's not a specific organ, it's the entire body. So another well-known autoimmune disease is type 1 diabetes. You know, the kids have to do insulin injections. So this is an organ-specific autoimmune disease. The pancreas is being attacked. Here, lupus is systemic, so the entire body is attacked. Its heterogeneous presentation means that the symptoms between patients vary a lot. So some patients have kidney problems, some patients have cognitive brain problems, some patients have arthritis problems, skin problems, a combination of those things. So this means heterogeneous. Flares and remissions means that it's not always bad. They get bad, then they get better, right? So flare, feeling lousy and really exhausted. So there's a lot of fatigue in lupus. It's not a nice disease to have. And then most patients are treated with steroids, and steroids get people to have swollen tissue. They gain weight. So prednisone, this is what the Chipmunk is saying, take prednisone, they say you'll feel better. So there is a lot of side effects due to the treatment. So now in terms of a little more scientific, what lupus is, this is from the NIH side. So one of the institutes at the NIH is called the National Institute of Arthritis and Musculoskeletal and Skin Diseases. So a lot of lupus research is being funded by NIMS. So the NIMS side said that the immune system is designed to attack foreign substances in the body. If you have lupus, something goes wrong with your immune system, and it attacks healthy cells and tissues. And this can damage many parts of the body, such as the joints, skin, kidneys, heart, lungs, blood vessels, and the brain. The prevalence, meaning that how many people have the disease, is between 40 to 50 per 100,000, so it's not that common. There is a very large difference between men and women. So the ratio of women to men is about 9-to-1. And in certain ethnic groups, it is higher than others. So in this country, the highest incidence is with African American women. So the most common lupus patients are young African American women. And it typically starts in the late teens, early 20s. There have been major improvements in treatment with immunosuppressive drugs. So in 1954, the survival at 4 years was 50%. So that was, in 4 years, you had 50% chance of being dead. Nowadays, it's less than 3%. So it's much better in terms of survival. However, it's still a very nasty disease. There is no cure. It's just making the symptoms less important. And there is also significant morbidity. So it means that people really feel lousy with this disease. And as we mentioned with prednisone, there is serious side effects from the treatment. The American College, so physicians taking care of lupus patients are called rheumatologists. Rheumatologists take care of also patients with arthritis and a number of more rare diseases. But so rheumatologists are physicians taking care of lupus patients. And so this is their society, and they have defined a number of criteria to diagnose lupus patients. So as I mentioned, there's a lot of variations in what the symptoms of those patients are. So there are those criteria, and I'm going to go over some of them. So the rash, this is called a mala rash. So you see this child has this across the nose here rash, which is usually triggered by sun exposure. And this is where the name lupus comes from. Lupus in Latin means wolf. And so in the ancient time, people thought that this rash makes people look like a wolf. So that's lupus means. Discoid rash is another type of rash which is all over the body. This is an example of thrombopenia. So you have swelling due to platelet problems. So you can see that there is swelling in the leg. There is also problems with arthritis that can occur. So the bottom line, it's a complicated disease to diagnose. And when you meet with patients, which I'm one of my funding sources is a society called the Alliance for Lupus Research, which is funded through donations from patients and people who want to support patients. So every year we meet with patients. And the striking thing that most of them have to say is that how long it took for them to be diagnosed with a proper diagnosis. So it's a complicated diagnosis, and people go from one doctor to the other before they get the right diagnosis. So why do we study lupus? So in 2001, there's this new drug, Benlysta, which has been approved, FDA, which is the Food and Drug Administration, which is a federal agency that reviews new drugs and approves them for use in patients once they're proven to be efficacious. So this new drug was the first new lupus drug in 56 years. And it doesn't work very well. So the standard of care treatments are either not very efficacious or have major side effects. Lupus is a disease in need for a cure. And so this is what we call the translational part. We want to find new therapies to help patients. This is a little bit of a jargon here, meaning that lupus is a good model to understand how the immune system goes wrong in attacking itself, right? So a lot of us are doing what we call mechanistic studies, so we want to understand what goes wrong in lupus patients. And by that, we understand better how the immune system works. So in my lab, we are trying to find the genes that are involved in lupus. So what are the genes that have mutations that cause people to have lupus? And because it has been shown that there is a genetic basis for lupus. It's not a simple genetic basis such as, for instance, cystic fibrosis, where there is one gene. If you have a mutation in this gene, you have the disease. If you don't have the disease. This is much more complicated. There are many genes, and some of them. So we have found some of those genes. Now that we've found some of those genes that are mutated in lupus, we are trying to understand what they do. And it's not obvious once you have a gene, to understand what it does. And so we start with a mouse, and then once we found what they do in the mouse, then we get blood cells from patients, and we are trying to figure out if the mouse gene does the same thing in human cells that it does in mouse cells. If it doesn't, it's not good, right? Because we don't want to cure lupus in mice. We want to cure lupus in patients. So we have to understand. We start with a mouse because it's easier to work with. And then we try to validate our results in people. A new area that we are getting into is the microbiome. You have heard what the microbiome is? I saw one head bobbing here. No? What is the microbiome? Have you heard that you have stuff in your gut that is important? Yes. So we are a giant colony of microbes in our gut, in our skin, in our nose, ears, everywhere, right? And those bacteria are doing a lot of things. Most of the time they're doing good things. You all know that if you take too much antibiotics, then your gut is not going to feel very well because you kill all of those bacteria. But there is more and more evidence that the microbiome regulates the immune system. So we have some evidence that the microbiome in lupus is different from the microbiome in normal people or normal mice. So we are trying to understand how that works. So we do a lot of poop work in the lab, mouse poop. So I'm going to finish by showing you not results, but type of techniques that we're using in the lab. So we do a lot of mouse work because we have mice that develop lupus spontaneously, and working with mice, you can ask questions that you cannot ask in people. You can treat mice, you can do a lot of things that you cannot do in people, of course. You can do experiments that you cannot do in people. We also get blood samples from lupus patients and healthy controls. So from those blood samples, we purify the immune cells, and then we do experiments in test tube with those immune cells. We have an instrument that is called a flow cytometer that is a standard instrument for to do immunology. So this is an instrument that allows to look at cells based on the protein that they have on their surface. So we have antibodies that are, I'm going to go on this side. We have antibodies that are labeled with fluorescent markers. And then, so we know, for instance, I'm going to give you an example. If we are looking at here, a type of cells called dendritic cells. So dendritic cells express this marker called CD11C. So we have an antibody against this marker that's going to be labeled blue. So then we pass those cells through this machine, and there are lasers that are detecting those colors. And based on the colors, they put them in different bins, and then we have computer programs to analyze those things, so we can detect in this patient or in this mouse, these are the dendritic cells, these are the B cells, the T cells, and so forth. So this is a very important instrument to do immunology. So we are using a lot of that. We also do tissue culture. We culture immune cells or stem cells. And we can do, here we are measuring growth, we are measuring differentiation, so we are taking stem cells and culturing them with different stimulation conditions, and then making, so here we made fat cells out of the stem cells and so forth. So this is the kind of thing we are doing. We are doing gene expression comparison between those cells. We are looking at metabolism. So metabolism, this is a cartoon of a cell showing that it consumes oxygen in the mitochondria. It consumes sugar here to make ATP. So we have a machine to measure this because this dictates how the cell is active. And then we can compare. So we know that in lupus, the bad cells that attack the body have a very active metabolism. So we are measuring these components, and then we treat the cells with drugs that makes metabolism slower, and then the cells are not autoreactive, they're not attacking the body anymore. So we are working a lot on that. We do microscopy. So these are T cells viewed with a scanning electron microscope. And we're interested in mitochondria here, which are the energy source of the cells. We treat mice with drugs. So in that case, it's metabolic inhibitors. So this is something to block glucose metabolism. Then here we, of course, take the organ at the end of the mouse when we sacrifice the mice and compare. We do some histology, we look at the kidneys, we look at their blood cells and so forth to try to determine what type of effect the drug has. So for instance, in this case, controls here, mice were not treated with anything. These mice were treated with this combination of drugs. They started here, this is bad, they get better. Their kidneys here are bad, and here they get better, so we say this drug is working well. And actually, in a couple of months, I'm going to China, where they are starting a clinical trial to use this drug in patients. We also do a little bit of molecular biology and genomics, so we're looking at gene expression. We are also looking at chromatin accessibility, so this is a little more complicated, but this is the idea that chromatin, this is how the DNA is packed in the cell, and depending on how accessible it is, based on methylation, acetylation, and so forth, you have more or less transcription. So we are doing some work into looking at that. And finally, as I was telling you, we are also looking at the microbiome in mice, and this is from something I found on the internet. We are only 10% human, so if you count all the cells that the human body carries, only 10% of those cells are human cells. The rest are bacteria that we carry. So if you think about that, those bacteria are not just innocent passengers. They are doing a lot of things that are most of the time good for you. I'm going to finish with two slides with recent undergrad projects in the lab. So Yunfei graduated from UF now. She was part of the UF minority program. She was minority because she was the first in her family to go to college. She did a very successful research project in my lab. She was working on a molecular project trying to figure out how to measure the expression of this complicated gene. Now she is in India doing a global health gap year project. I think she's going to be there for 2 years with an NGO while she's applying to medical school. And she sends a lot of emails trying to get money for her project. And then this is Hope Summers, who is a current Santa Fe 2 UF Bridge student. So she started last summer. She has done a research project over the summer that she presented as a poster at a meeting, I think it was in Tampa. Yeah. Yes. So we are breeding mice, and so she was in charge of detecting specific genes in those mice that we were looking at. So she's doing a PCR, which is an amplification of those transgenes in DNAs that we clip from the tail of mice that we want to identify. So this is one of the gels from the PCR reaction that she has done. And that's all I have for you.